Okay, please let me know if at any point you're unable to see anything on the screen. So I'll start off with the discussion for today first. And the reason I'm not doing a recap of the previous session is because after today's discussion, which is going to be pretty brief because we didn't have a lot of topics left. So we're just going to learn a little bit about cancer statistics and some terminologies that are very commonly used to represent data. And then we'll go through a quick recap of the course. And in the next session, hopefully on Monday, we will discuss all of the previously. Just a second. I'll Yeah. Okay. Sorry for the disturbance. Yeah. So then we'll end the next session with all of the previous questions, like I've mentioned before. So majorly the topic that we're covering for today is understanding cancer statistics. So under that, okay. So first of all, I want to introduce you to the concept that whenever any study is carried out for a population, then the idea is to present the data for that population in a particular manner. So we'll first understand this topic and then I'll take you through a couple of examples of some research papers that'll probably help you understand it really well. So yeah. So basically, whenever we talk about cancer statistics, there are three terms that are very commonly used. The first one is cancer incidence. Now, while we know the layman meaning of what incidence is, which obviously means something, an event which has occurred, right? But whenever you talk about terminologies like these in context of any disease, in this context of cancer, then it has a specific formula attached with it. It has a particular definition which is attached with it, and therefore these terminologies should not be used loosely in context of any disease. Why so? That is because these statistics or these numbers have been arrived at as a result of a very extensive study that is carried out by certain organizations. Like for example, here, if you look at this image shown here, the data that I'm familiarizing you with today is from Globocan 2022, which I think I had explained in one of the previous classes, but I'll take you through the website again today as well for your reference, just in case somebody missed the previous discussion. So, Globocan 2022 collects data from different sources. One of them is IARC, which is the International Agency for Research on Cancer, and that is managed by World Health Organization. And this IARC also collects data from smaller entities, which include NCRP, etc. You can read about these. Like there are extensive papers if you want to learn more about how exactly all of these statistical, how these numbers are exactly arrived at. So you can refer to that. But basically, that's what I wanted to familiarize you with. That every couple of years, like for example, this is the most updated data you'll find on Globocan because the last stats are from 2022, but that gets uploaded almost 2 years later. Like in 2024, this was uploaded. So that's why the information you are getting is like, for example, if somebody does a stats, an information collection today, collating all of that information and its publication is going to, of course, take a little more time. So therefore, whenever we report anything in any publication, any journal, writing these terminologies has to be done with a lot of caution, and whatever numbers you are citing in your paper, whatever you are providing information, that has to have proper references attached with it. Now Globocan is a very reliable source, so whenever you have to say draft a thesis, draft a project or a review, whatever is your objective of interest, then you can certainly refer to this. And along with it, if you go to the website, I'll show that as well to you, you'll find the papers from which it has also been correct, like added. And so those information should also be a part of your document. So when we talk about cancer incidence, it basically means the number of new cancer cases that arise in a specified population over a given period of time, typically in 1 year. It can be expressed as an absolute number of cases within the entire population per year or as a rate per 100,000 persons per year. So a standardized rate is something we'll come to later, but basically this is what it means. So incidence rate comes from the number of here new cancers basically means the number of people who have been diagnosed with cancer in that particular year or in that particular period of time, which is typically 1 year, for that particular population into 100,000. The numerator of the incidence rate is the number of new cancers. The denominator is the population size. The number may include multiple primary cancers that are occurring in one patient. The primary site is reported as the site of origin and not the metastatic site. In general, incidence rate does not include recurrence or relapse. It means a new case. That means a person who has been diagnosed with cancer for the first time will be included in the count for a new cancer incidence. The population used depends on the rate to be calculated. For example, for cancer sites which occur in only one sex, the sex-specific population is used, like for example, females for cervical cancer or prostate for male cancers, etc. However, the graph that you see here on the left, you will find that these are the applied parameters which I used when I extracted this data. So this is for absolute numbers. This is showing you absolute numbers, like you can see approximately, I think, 24 lakhs 80,675 cases of lung cancer has been, which ranks it as the highest, as the cancer with the highest incidence as per 2022 in both sexes, right? However, if you put a sex bias there, if you wanted only males, only females, you would find that information as well. Also, on the portal, we'll come to it, you can also figure out the most prevalent or most common cancer in a particular population also instead of the site of cancer origin. Now, I had just very quickly mentioned the word prevalence, and that is often very commonly used when we speak just in regular English. A lot of times incidence and prevalence are used interchangeably. However, you have to you have to understand that there's a very key difference between those two terminologies, especially in context of cancer. For that reason, here you'll see that prevalence, or in this case, cancer prevalence, is mentioned very separately, and it's a completely different statistic used to explain the abundance of a particular disease in a particular population in a particular period of time. Prevalence, on the other hand, is defined as the number or the percentage of people who were alive on a certain date in a population who previously had a diagnosis of the disease. So it includes the number of individuals within a defined population who have been diagnosed with that cancer and who are still alive at that particular point in time. But it includes new cases and pre-existing cases and is a function of both past incidence and survival. So it includes both types of cases. This makes it different from the term incidence. Keep in mind, incidence means a new report. Prevalence includes those who were diagnosed with cancer previously. And like, for example, if we have the prevalence of a particular cancer today on the 28th of March 2025, then of course that will include any individual alive who have this particular cancer. Say, for example, let's talk about lung cancer. The number of people on earth who have been diagnosed with cancer on this date will give you the prevalence of the disease. And that includes both cases of past incidence and survival and cases which have been reported freshly in the last couple of months. So when you take prevalence into factor, you can see at the top, you can find breast cancer to be the most prevalent disease. Again, go back to the previous slide and you will see that lung cancer had the highest prevalence, followed by breast, colorectal, prostate, stomach, and liver cancer. However, here you can see that breast and colorectal overtake lung cancer. Prostate is pretty much around the same place. Thyroid cancer emerges quite towards the top. You also have stomach cancer, and other cancers include a lot of other different types, which you can refer to. And this is indicated in this way because otherwise you would not be able to show so much information on this graph. But in a tabular fashion, all of this information can be availed very easily. So this is the difference between the terms incidence and prevalence. The third thing that you should also learn about is that whenever you talk about cancer, the most common way in which people try to highlight the importance of their study or the importance of their research is by highlighting A, how common that disease is, and B, how much of an impact that disease has on a particular population across the world. So to explain that impact, obviously, oh, I think I put the wrong graph here by mistake. Just a second. Yeah, it's the incidence graph. Wait a minute. I'll show that to you. Okay, I'll show it to you from the portal then. Yeah, I have accidentally put the wrong graph. But anyway, let's understand the definition first. So, cancer mortality rate is the number of deaths with cancer as the underlying cause of death that occurs in a specific population during a year. It is usually expressed as the number of deaths due to cancer, again, per 100,000 people in a population. So this is the statistic used. Mortality rate is cancer deaths per population into 100,000 number. The number of the mortality rate, the numerator of the mortality rate is the number of deaths. Denominator is the population size. Population used depends on the rate that you want to calculate. And again, the same statistic applies here as well, that if you have a gender-specific or a sex-specific cancer, you should only use that population for consideration as well. And also, mortality rate can be given or can be calculated for one particular site or for all of the cancers combined as well. Another statistic which you should learn about, why you should learn about it, is because these numbers that you see, they have been derived from the age standardized rate, which is ASR. Okay, now what exactly is ASR? Let us understand that. ASR, or age standardized rate, gives you a summary. A summary of what? A summary measure of the rate that would have been observed if the population had a standard age structure. Standardization is necessary when comparing multiple populations that differ with respect to age, because age has a strong influence on cancer risk. So an age specific or an age standardized rate is a weighted mean of the age specific rate, and the weighting is based on the population distribution of a standard population. Most frequently used standard population is the W standard population, and then what you present is called as an age standardized mortality rate or an age standardized incidence rate for a particular disease across the world. So what does that mean? It means the parameter of age. Okay, 8.2 was off. Yeah. So the age standardized rate, or ASR, is a way of representing information across different populations. Why is it required? If you sorry, if you look at this graph, this is to show you the difference between, like, for example, let's look at it this way. Let's take the image one at a time. What is it showing you? It's showing you the ASR for the world per 100,000 individuals and the incidence of the of cancer in both sexes in the year 2022. What you can see, the first most obvious thing is, look at these little graphs. You can see these are the probably the Caribbean, and you can see these little islands, or if you go along this Indonesian belt, you can see little islands, and then you can see much larger countries. However, you also have to keep in mind that the land mass and the population number, all of these are very different. So whenever you make a report of a particular statistic, you have to take all of these differences into calculation. First of all, whether or not that particular disease has been studied for that particular population, whether its data is accessible, whether or not you are able to like include all of that information into consideration, all of this has to be taken into mind. So to represent these statistics, what is done is that for each population, a weighted mean is taken, which means that apart from the number of cases which are getting reported, you also um, include the number of individuals present within that population to balance it out, to normalize the incidence rate of that particular population, of that particular disease within that particular population, and then you represent that. You will see that there are certain countries for which no data is available. Even in India, you can see that some portion, particularly towards the north, towards Jammu and Kashmir, you can see that a lot of places data is not available or is not accessible. So based on all of this information, what is done is you consider the standard population, and with respect to that, you modify the rate of you modify the statistic and then you represent it. So whatever representation is done finally within a paper is all normalized, taking into consideration the number of individuals within that population, and then the rate of that disease is then publicly available. So that is the ASR. And apart from that, another statistic very, very commonly used for any paper which talks about cancer is the Kaplan-Meier survival curve. What is its job? It is defined as it's often known as the KM curve. It is defined as the probability of surviving in a given length of time while considering many small intervals of time. There are three types of assumptions which are made in this analysis. First, it is assumed that at any time, patients who are censored have the same survival prospect as those who continue to be followed. What does that mean? It means that if there are certain patients who have discontinued, um, say for example, if you have a clinical trial of 50 patients, mm, yeah, let's assume that's a very small number, but just for the sake of a discussion, say you are starting off with 50 patients, and among them, only 20 or 25 of them are turned up for your follow-up examinations, and the rest of them cannot be tracked. Then what happens is that the information, as long as you have, what is expected is that they have the same survival prospects as those who cannot be followed. If it is such that the number is very huge, like for example, if half of the patients have not been followed, then at that time, that information has to be omitted from your clinical trial, from your study, and then it cannot be concluded. Like, for example, if from a study of 500 individuals, if two patients cannot be followed, that is an acceptable information to be censored. However, the percentage of individuals, if it is too high, then that data it would be considered to be inconclusive or might even be considered to be insignificant. And then that study would not really be very acceptable in general in clinical setting. Secondly, it is assumed that survival probability is the same for individuals who were recruited early and late in the study, which means that from the end of the institute or the medical organization that is conducting that study, no changes have been made. So somebody who was enrolled, say, in January versus somebody who is enrolled in March today, they would receive the same way or the same parameters of the treatment which is being administered. And so whatever you are observing, all of that, like think of it as a race. It does not matter if somebody has enrolled in the first in January versus somebody has enrolled in March. If the race is starting, then you would all assume that they are all beginning from the same start line, and then the comparisons are normalized from that day onwards. So that is another assumption which is made for Kaplan-Meier curve. That is obviously necessary because in a study of 500 individuals, you probably cannot start the treatment for all 500 individuals at the same time. Why? Because the grade of the disease or the aggressiveness of the disease might not necessarily be comparable in all patients, right? So somebody might be diagnosed 1 month ago versus somebody might have been diagnosed 1 year ago. So they cannot be enrolled at the same time. So that becomes a practical limitation. And therefore, for Kaplan-Meier curve plotting, it is assumed that there is no effect of the time of recruitment upon the results of the study. Thirdly, it is assumed that the event happens at the time specified. This creates problems in some conditions where the event would be detected at regular examination. We know that the event has happened, and estimated survival can be more accurately calculated by carrying out follow-up of individuals, frequently at shorter time intervals, as short as accuracy of recording permits. The Kaplan-Meier estimate is also called as product list estimate. It gives you a probability of occurrence of an event at a certain point of time. So yeah, these are just additional statistics for you to know about the plot. How is the plot generated? So first, let me just quickly show you the plot. This is what it overall looks like. This shows you the cumulative survival of patients, and this is the time. So over a period of time, basically what is monitored is that how well or how many people are surviving and what is the cumulative survival of patients over that period of time. So for every few days or every X number of period of time, you can see some statistic has been plotted, and how a patient responds or how a group of patients respond to a certain treatment will help you conclusively understand how that treatment has worked. So how is this survival function plotted? Let us again go back to this slide. So for each time interval, the survival probability is calculated as the number of subjects who have survived dividing by the number of patients at risk. Subjects who have died, dropped out, or moved out are not counted as at risk. That is, subjects who are lost are considered censored and are not counted in the denominator. Total probability of survival till that time interval is calculated by multiplying the probability of survival at all time intervals preceding that time. So if you have studied basic probability, you would know that you would basically have to get the cumulative probability, you would have to multiply all the other probabilities at that preceding that period of time. Maybe I'll show you a easier way of understanding this. This might actually be very confusing. Um, okay, let's uh, looking for a clean example for you to understand, but I don't think it's very clearly mentioned. Let me get you the formula at least. Basically, this is what is done. Survival at time t and survival at time t minus 1 are both factored in to give you the effect of that particular treatment or the effect of that particular disease. So I could not find a very good resource for this, but it's okay. You can, yeah. Basically, this is what happens. Let me zoom in a little bit. So this is the statistical function that is used to calculate the probability of survival. So the probability of survival at time ti is what you're calculating. For that, what you do, you take the probability of surviving at time ti minus 1 and you multiply that with 1 minus number of patients surviving before time t, which is ti in the denominator, and in the numerator you have number of people that have the event of interest, which means that are being taken into consideration at time ti. So all of that is together used to measure the number of people who have that particular disease at the time of interest that you are studying it. So this has been simplified and represented here. Overall, for each time interval, what you calculate is the number of people who were living in the beginning, number of people who died, a number of people who were living in the beginning is used in the denominator. Altogether, these functions are used to calculate the survival function, which is what you're seeing in this green plot. And from here, the red shows people who have discontinued, they are censored individuals, and that is used for your calculation purpose. Here, the example given is for the probability of a patient who survived 2 days after a kidney transplant. They can be considered to be having the probability of surviving that particular day, day 1, multiplied by the probability of surviving the second day, given that the patient survived the first day. This is called as conditional probability, which is all taken into consideration. Because of the small number of events which are all together getting integrated or basically summed, the overall probability of surviving to each point becomes more accurate. So what you effectively do, you take the probability of surviving day 1. Next, you multiply it by the probability of surviving day 2. Having survived day 1 makes it a cumulative probability. You add that you add day 3, day 4, day 5, so on and so forth. And then gradually the probability function gives you a curve like this. And in comparison, when you take the other condition, if there's a difference between the two, say for example, there was another line which showed you that the probability of survival would otherwise have been very high and your treatment is compromising this thing, then that means your treatment is not good and vice versa. So overall, that is the information I wanted to convey to you. I'll show you a different representation from a paper that should help you clarify things. Let me just share my screen quickly. Yeah, so this, what I'm showing you right now is a paper from my lab. You don't have to focus on other things, but basically what they have tried to show in this paper is that the high expression of this particular gene leads to very poor prognosis of patients, and it has a huge role to play in disease progression. Basically, it helps in formation and progression of the cancer. So to explain that story to other individuals, what you do is you use information which is already publicly available. So in this case, that public information, you remember very early on, I discussed with you about TCGA, which was the Cancer Genome Atlas. So from that Cancer Genome Atlas, you can check what is the expression of your particular gene. This shows you, the y-axis shows you gene expression. And you can see that in red, you can find the expression levels of that particular gene in a normal individual versus the expression of that gene in individuals with cancer, which shows you that it has high expression in cancerous patients or in individuals diagnosed with cancer. Same statistics can be checked in different stages of cancer. In a benign, localized versus metastatic disease, you can see that the disease expression, gene expression increases, which further helps you explain your story to other individuals that this gene is associated with disease progression. Third is what we are more interested in. Let me zoom in a little bit. This is the example of a Kaplan-Meier curve. What it shows you is the probability of survival of individuals over a period of time. Now you can see that high and low expression of this gene and the impact of that on survival of patients. So in blue, you can see that when individuals have low expression of this gene versus when they have high expression, higher expression leads to poorer survival, which means that the disease has led to more poor outcomes. That means, not the disease, high expression of the gene in the disease had led to more poor outcomes, and the probability of survival of patients is much, much lower. So this is how you draw a comparison. Apart from that, you also pitch in the idea of the expression of your a gene overall in the world. If necessary, you can check it in context of cancer. You can see how abundant that cancer is. So all of these different statistical terms are used to explain or to validate that whatever study you are carrying out, is it really relevant to patients? Is it going to be translatable? For example, you are seeing a particular gene's expression in a particular cell line, but what is the impact of it? What is the role of studying it? For that, you have to see how does it affect patients across the world, and then you can figure out a way to represent that using all these different terms, like I explained: incidence, prevalence, mortality, and finally the survival curves. So if there are any doubts about this, please let me know. I will use some other information to explain it to you. And in the meanwhile, I'll open up the Globocan page. Any doubts, any queries? Okay. So yeah, this is the repository that I was talking about, which gives you all of these information as up to date as possible. I'll also just um, to figure out where this information is gathered from, you can search cancer statistics. Yeah. So in cancer statistics, you'll find all of these different information. So you'll find a lot of details which are given. Um, just a second. Statistics 20. So the last report was published in 2024. So yeah, you can see the number of citations that will explain to you how relevant this information is. So you'll find this journal, you'll find this issue by Siegel et al. And most of the time, this information is free, so you can use it for your reference. And there you'll see all this information. What is the source of the data that they use? How there are collaborations between multiple institutes that are running this year program, and which locations are they using for sampling? What is the way in which they are showing all of these different reports, like, for example, incidence, mortality, like I just discussed. Furthermore, they also give you information about projected rates of that disease. So based on previous statistics and based on the current progression in the disease, they give you projections about how far the disease is going to progress in the next 20 odd years. So all of that you can also obtain. Let me show you the one from India. Yeah. So this was published in the year 2020 from National Cancer Research Program, or NCRP, in India. So that gives you the information about the condition of cancer in India in the present scenario. So basically what they do is they calculate all of these different parameters, incidence, trends, projections, mortality, etc., from different PBCRs and HBCR. PBCRs are population-based cancer registries. Hospital-based cancer registries are HBCRs, and they have all been associated with the NCRP for a very long time. So from there, they get all of this information. I'll show you these tables. So yeah, if you look here, you'll find information about the average number of patients with their incidence rate per 100,000 in different states. You'll find a sex-wise separation. You'll find overall cumulative risk. You'll find mortality. And it also takes into factor the sex bias, so that has been represented separately. Apart from that, you can see this representation of age-adjusted incidence rates or age-standardized incidence rate as well. And you'll see that in males, it is quite abundant in the Azol district. It's quite prevalent. Cancer is very prevalent there. Then district-wise separation also you can observe. Yeah, basically there's no point in going over all of this, but yeah, I just wanted to show you a few statistics so you're familiar with it. This is the projected incidence of cancer statistics in India. And as you can see, you'll find that a separate comparison has been drawn for males, females, and individuals from both sexes, and the number of cases which have been reported for all sides versus different segregations have been done. Also, you'll find certain regions to be grouped, like, for example, the digestive system overall is also represented in a lot of places. You'll find respiratory cancers grouped together. You'll find urogenitary and urogenitary cancers often grouped together, endocrine cancers grouped together, blood cancers grouped together. So all of this you can also find in a lot of places. So basically that is how all of this information is collected, and it is all in collaboration with ICMR, or the Indian Council of Medical Research. So taking into factor information from many countries across the globe. I just showed you the example of India, but collating all of that information together, it is then published in this repository that I might have introduced you to before, which is Global Cancer Observatory or GloboCan. And you can read more about it here for your own reference. But basically, what we normally use it for is for finding out a quick schematic representation of all of these different statistics. So among them, you can see cancer today, which is the one I most commonly used, and the information I've shown you today is from there, which means it gives you the current status versus cancer over time, cancer tomorrow, causes, survival, and all of that information is used as well. Um, Sorry. Cancer Tomorrow is going to show you projections of future cancer incidence and mortality burden worldwide from current estimates made in 2022 up till 2050. Cancer over time shows you how it has, what kind of a trend it has followed um, over a period of time and what it is expected to be till the year 2040. And of course, this gives you most of the information that I showed you today. I'll open this up and show you the mortality fact because I missed out on it. So yeah, these are all of the different tools. You can use bar graphs, pie charts, heat maps, scatter plots, or tables. Most commonly, you'll find either bar graphs or pie charts used for representation because they give you a lot of information within a small space. These are all of the different fact sheets. You can use any of this for your understanding. So based on cancer sites, all of the information, maybe let's quickly go here and then we'll come back. So this is giving you the burden of cancer in a selected country or a selected region for each cancer type. And you can see how certain cancers have been grouped together here as well. Some female cancers have been separated, male organs have been separated, urine cancers have been separated. All of this is categorized based on countries, based on UN regional groups, or based on populations. You can find different statistics. So yeah, that's all of the information. Let's go to pie chart, the most commonly used representation and the one I showed you as well. So the default loading that you get is a representation of the incidence of cancer in different populations for both sexes across the world, right? So among these, you can find that in Asia, approximately half of all of the reported cancer incidents can be found in Asia itself. Yeah. Apart from that, you also find Europe, Northern America, etc. to be present. You can modulate these. First of all, you switch to different cancer types. I think this is the one I showed you before about cancer incidence. Here you can switch it to mortality, and you can see there's a slight shift. Prostate goes down. You can find this to be quite abundant, and yeah, I think this is stomach. Yeah, stomach cancer comes up, and next it is followed by pancreatic cancer. Here you get other information also. You can get rid of specific populations. Um, you can go for a male specific. Uh, why is this not adjusting? Just a second. I think it's sort of stuck. Yeah. So when you come to male cancers, you'll see a different grouping. For female cancers, you'll see another grouping. Moving on to mortality, we were looking at mortality and both sexes. Among these, you can see that lung, colorectal, liver, breast, and stomach and pancreas, they mainly predominate. You can also modify this with the number of slices. Say, for example, the cancer you're working on is lung cancer, so that you can highlight this a little better. That lung cancer is the most abundant cancer, has the highest incidence. Apart from that, yeah, these are just beautification purposes, but yeah, not really that relevant. Apart from this, you can compare different populations for different types of cancers. This is with respect to mortality. You can compare both incidence and mortality as well, like you can see. So if you have, been in contact with a lab, or if you worked personally as well, you must have seen papers statistics and graphs like these used for representation very commonly. So it is good to know how to retrieve all of this information. See, this is all publicly available. You just need to know what is relevant to you, right? Also, here you can adjust age. For example, if you see that a certain cancer is more prevalent in a certain age group, like for example, if something is a very late onset disease or something is a very child specific disease, so you can adjust that also very, very easily. So yeah, these are the different, these are bar charts, population comparisons, pie charts. This shows you a heat map. Um, yeah, this is the heat map, which I showed you previously, I think, for incidence of cancer. You can also get tree map representations from here. So basically, the size of the box indicates how prevalent that particular cancer is. And you can develop it differently, like hubs or human development index, etc. You can use one particular type of cancer. All of that is like for you to completely explore. Um, this shows you prevalence, which is the other statistic that I talked about. So yeah, you can use it for your own understanding however you want. So yeah, that is overall what I wanted to.
तीन तरह ना अच्छा ये वाला ये
Ankit,
पर अभी तो
you
मैं
need to mute yourself. Okay. So yeah, overall, that's what I wanted to explain about cancer statistics to you. Uh, any doubts there? Otherwise, I'll start the course recap and then we'll pretty much be done with all of the information we wanted to cover for the course. And we'll address the queries in the next session. So if there are any doubts, please let me know. Okay, so I'll start this recap. If you've already attended the sessions, you're already familiar with it. Um, if there are no doubts, it is fine, but I wanted to give a summary before we address the questions for this because this was a very long course. So I don't think I'll be able to wrap up the entire summary by today. So I figured we should start the recap today and then spill it over to the next session. But basically, what we had started off with was an introduction to the basic concepts of cancer. So we talked about how cancer is a disease of uncontrolled cell proliferation, and we discussed this as to be the most commonly known and the most common perception of cancer, that cells divide very abruptly and they generate very large masses of tumors. We talked about, sorry, we talked about two, or we actually brought about two questions. First is, do cancer cells transform? And second, do cancer cells evolve under natural selection? This question we addressed very early on in the course about whether cancer cells actually transform or not. The natural selection is something we addressed very recently, where we learned about how cells evolve under drug pressure, how there is a transformation, or not a transformation, but transdifferentiation of cells from one lineage to another when the surrounding environment is changed, when there's a modification or there's a selection pressure by some sort of a drug, which causes the cell to either change itself or from a population of cells to bring in the Darwinian idea of selection, and only survival of the fittest is permitted forward. So the treatment resistant clones are the ones that are able to survive. So we addressed these later on, but what we started off with was the revolutionary idea. Not even an idea, but basically they collated information that is already available and they presented it in the form of key characteristics or key features, which they defined as hallmarks. So these amazing scientists were Professor Douglas Hanahan and Professor Robert Weinberg. So they introduced us to the concept of hallmarks, which we'll come to later. We had first understood the different substeps within the progression of cancer, starting off with initiation, hyperplasia, dysplasia, in situ cancer, invasive cancer, and finally metastasis. Among these, we had looked at how cancer progresses, and we had seen that from a normal epithelial cell or from a normal or physiologically functioning cell, when there is a mutation or any sort of genetic disturbance, or there's an insult to the genome of the cell, then it undergoes modifications or alterations which cause it to divide very haphazardly, leading to the formation of this abnormal mass. But yeah, again, keep in mind abnormal in the sense that it is not expected to be present there. So these are just more number of cells that have been formed as the result of very rapid division. So that is called as hyperplasia. Following that, when you have within that particular mass, when you have a subpopulation of cells that have very increased growth potential and that show abnormal tissue architecture, then that you consider to be dysplasia. Following that, when cells are forming a huge mass and they continue to behave in this particular manner, then that leads to in situ cancer. Following which, you can find that there is a if yeah, if there's the transformation of these cells to be able to invade the particular tissue where it is residing and then move into the blood and the lymph of the individual, then that would be considered to be invasive cancer. And then that invaded or those invaded cells are able to move through the vasculature of that individual through blood vessels or through the lymphatic vessels, and they're able to metastasize to other sites where they would form secondary metastasis or secondary tumors. And this is the result of several sub-step or sub processes as well, which include invasion, intravasation, dissemination, extravasation, and colonization. Now, I think during the class, I had not focused a lot on these subtle differences between these. I think we did not discuss it. So I'll go over this in a little more detail, and I think it's one of the questions, takeaway questions from this class as well. So let us understand the key differences between each of these processes. So after this mass of cells have been formed, what first happens is that there is the breakage or the disruption of the basement membrane. Basement membrane is the support or is the substratum on which this entire tumor is residing. So they break this. How do they do it? They do this because there is a link between the so two things have to happen. First of all, cell-cell adhesion has to go away, which means the intercellular contact has to go away. And the second thing is that the cell and matrix contact that has to go away. So that goes away by the use of certain proteins known as MMPs or matrix metalloproteases, which are, as you can understand, they are metalloproteases. They are proteases and they are degraders of proteins. They chop proteins and they use a metal for their activity, which leads to their name. So these MMPs, they disturb this integrity of the basement membrane, and now cells acquire the ability to break that membrane and move across it. That process is invasion. Just still there, the ability to disturb or disrupt the basement membrane and breach it and move across it is known as invasion. Following this, what do? They move into the nearby tissue, right? Following this, they undergo the process known as intravasation. What does that mean? Very different, like it's easy to understand. It's a very direct nomenclature. Intravasation, as in it is inside some sort of vasculature, right? Vasation means there is a vasculature which it is entering. So these cells are then able to move into the vasculature very close by the process known as intravasation. Following which they dissociate. Like for example, if there's a mass of cells that are moved in, what they do is they disseminate, which means like basically they sort of become individual cells. Why? Because it's easier for them to move. Like imagine an entire mass of tumors of, say 50,000 cells. That will not be able to move through vasculature, right? Our arteries and vessels don't have that much space. So what happens? They undergo the process of dissemination, which means that they are able to dissociate it and as individual or smaller clumps of cells, they're able to move through the vasculature. Following this, it moves to its site of interest. Say, for example, this was site 1, and now it has moved into site 2, which is providing the tumor cells a very comfortable and very pro-cancer environment, which means the microenvironment there is favorable for the cells to be able to grow. So in that environment, what do? They then undergo the process known as extravasation. as the name suggests, is the exact opposite of the process it had undergone previously. So now what they do, they move out of the vasculature and they then move into the surrounding microenvironment or the surrounding tissue environment. And there they form the mass of cells by a process known as colonization. Colonization or homing is the process where cells again start, associating, reassociating. Why? Because they underwent dissemination previously. So now they again have to gather together. They again have to form a mass of cells, which will then inhabit within that region, and they'll be able to form secondary smaller tumors. Now understand that from the entire population, say if 20% cells, for example, had invaded and had undergone intravasation and moved into circulation, from that 20%, only maybe 1 or 2% might have localized here. The rest of the 20%, around 17 to 18%, can then move anywhere within the body where they find a very suitable environment, and there they can form new secondary tumors. That is why metastatic cancer becomes very, very difficult to treat, and its diagnosis also, it's it's more favorable to have earlier diagnosis, because in those circumstances you would have smaller tumors that would be easy to find or to detect, and then obviously to treat. So yeah, that's what. Okay. So these are the different substages of tumorigenesis. Then we understood these differences between key terminologies in cancer and different types of cancers. So hyperplasia, like I already explained, is a stage where cells are able to multiply very fast, much faster than normal, and extra cells, they build up. However, the tissue organization, it appears normal. There is no there is no disruption of the tissue architecture. So it looks normal. It's just more number of cells within that particular region. Second was dysplasia, more advanced than hyperplasia. There is also a buildup of extra cells which appear very abnormal, and the tissue organization is now altered. An example is an abnormal mole, also often called as a dysplastic nevus, that forms on the skin. So this, you can see the main difference that I had mentioned at that point of time, if you remember, was that from the initial architecture of cells, in the dysplastic cells, you can see that the size of the nucleus is way more huge. And the nucleus to cytoplasmic ratio has been completely disrupted, as a result of which that this tissue is now almost almost indefinitely going to progress towards cancer. So that's what you see eventually, that there's development of cancer. Second, this was a comparison between normal skin and epidermal hyperplasia. As you can see, there are these patchy regions where very rapid cell growth has occurred, and these cells are all abnormal in nature because of the size of their immense size of their nucleus. And obviously, as you can see, compared to normal skin, the tissue architecture is also significantly disrupted. Then we had looked at this comparison between low grade and moderate to high grade dysplasia, and finally carcinoma in situ. And we saw how over time we can see that there's a gradual increase in those two characteristics, the loss of tissue architecture and the huge nucleus to cytoplasmic ratio, and the abnormal organization overall within the sample that has been studied. Then we understood all of the different subclassifications of cancer, like tumors can be classified into broad categories, mainly benign versus malignant. Then based on the source of origin, we understood how classification can be done further, starting off with the definition of carcinomas, most common cancer to be diagnosed in patients, which arises from epithelial cells covering the interior and exterior surface of the body. We saw this comparison between carcinoma in situ and cancer as well. So carcinoma in situ meant the site where the tumor has formed. So these cells, these abnormal cells, they were found only in the place where they first were forming, that carcinoma was formed within the body. These abnormal cells can potentially become cancerous later on and then spread to nearby normal tissue. So that is what you see in carcinoma in situ. You can see that the membrane is intact. It has just like even your membrane is intact, but basically you can see that it has not disrupted the structure too much. It is still a part of the membrane and has kept the overall architecture okay. Whereas in cancer, you can see that a huge mass has formed, which is obviously not expected and very haphazardly grown as well, which is indicative of cancer. Um, Sorry. Okay. Then we understood the different nomenclature under carcinomas as well, like adenocarcinoma forming in epithelial cells that produce fluids or mucus, which is majorly generated from glandular tissue. Then basal cell carcinoma that begins within the basal layer of the epidermis within a person's skin's outer layer. Squamous cell carcinoma in squamous cells present within stomach, intestines, lungs, bladder, kidneys, etc. Transitional cell carcinoma, which forms in epithelial tissue known as transitional epithelium, which is made up of many epithelial cell layers. Usually, they are found in the bladder, ureters, part of kidneys, few other organs, etc. This was a comparison of all of these different types: colorectal, basal, squamous cell carcinoma, and we had looked at this last time as well, that for transitional cell carcinoma, what are the different sub categories that are necessary for characterizing it as a TCC or a transitional cell carcinoma. First of all, there is a loss of nuclear polarity, which means that cells do not mature towards the surface into umbrella cells with low NC ratios and abundant cytoplasm. Okay, just a second, sorry. Yeah. Uh, yeah, so cells lose their nuclear polarity. They exhibit large, irregular, very hyperchromatic, as in very darkly stained nuclei as well, making them very characteristic of TCCs or transitional cell carcinomas. Then we understood the meaning of sarcomas, which form in the soft tissues and bones. Um, Muscles, fat, blood vessels, lymph vessels, all of these are under the purview of sarcomas itself. Then we understood examples of osteosarcoma as the most common bone cancer. Other types of examples were also cited. You can see two key examples of soft tissue carcinoma and Kaposi sarcoma indicated in these images shown. But apart from that, other types of sarcomas are also known, like angiosarcoma, osteosarcoma, Ewing's chondrosarcoma in cartilage, GI stromal tumor, fat cell based liposarcoma, the fibrous connective tissue sarcoma, etc. So all of these are different subtypes. Then we understood what is leukemia. Leukemias are cancers that begin in the blood forming tissue of the bone marrow. They do not form solid tumors. Instead, there is an increase, or there is a very large production of abnormal white blood cells, which are leukemia cells and leukemic blast cells. They build up in the blood and they crowd out the normal cells as well. I think I'd given you a better example of leukemia versus lymphoma. But basically, what we saw is that there is a huge production in, or a very aberrant and very abnormal production of these particular cells within the bone marrow of these individuals. And the excessive production is a problem because it basically competes with normal cells of the body. And as a result, you would have lesser and lesser resources. It would be more difficult to control bleeding because at that place, platelets would not be able to, arrive. And fighting off infections might also become a challenge. Then you see for lymphoma, it is a cancer beginning in the lymphocytes, the B and T cells. Abnormal lymphocytes build up in the lymph nodes and the lymph vessels as well as other cells of the body. There are two main types of lymphoma. One is Hodgkin's, one is non-Hodgkin's. In Hodgkin's lymphoma, people have abnormal lymphocytes called as Reed-Steinberg cells, which usually form from these I3 from these B cells. Then you have non-Hodgkin's, which begins in the lymphocytes. They can develop either from B cells or from T cells. Um, then this was a comparison between these two with myeloma. In multiple myeloma, it begins in plasma cells. They build up in the bone marrow and they form tumors in bones all throughout the body. It's often also called as plasma cell myeloma and color disease. Melanoma is a cancer that begins in cells that become melanocytes, which are necessary for making melanin. They form on the skin, but they can also form in other organs like the eye. Apart from that, we had mentioned that there are other types of cancers as well, like neuroectodermal cancers, including gliomas, neuroblastomas, medulloblastomas, etc., which arise from various CNS and PNS components, and germ cell tumors as well. Then we moved on to the hallmarks of cancer. Now, before we go into that, if there were any queries, please let me know from any part on this. Okay, moving on then. We have discussed hallmarks of cancer, majorly from these three reviews, like I mentioned previously. Please go through it. It will, if not anything, it will be extremely educational for you. So please go through it. You'll learn a lot. So what they defined hallmarks as were the features or the functional capabilities that are acquired by human cells as they progress from normalcy to cancer or cancer-like state. And under that, they had subcategorized these into majorly two domains. One was the core hallmark, and the other included emerging hallmarks as well as enabling characteristics. I think I had put this also as one of the questions to be discussed. So basically, the key difference between these two is in context of um, how well it has actually been established. I had suggested people to go through the third hallmark paper, basically the new dimensions. I think that was the third one. Um, Let me open it up. Let's have this discussion today itself. Just a second, please. Yeah. So if you open, this is the third paper called The Hallmarks of Cancer, New Dimensions. So within this, you'll see that they have mentioned that while these were the core hallmarks, which include the ones that we have discussed, sustained proliferative signaling, evading growth suppressors, resisting cell death, enabling replicative immortality, inducing vasculature, activating invasion and metastasis, and reprogramming cellular metabolism, avoiding immune destruction. These were added later on, but the core six hallmarks were added first. But what they have also talked about were the emerging hallmarks, which included the cellular deregulation of cellular metabolism and avoiding immune destruction that were segregated as emerging hallmarks in the previous concept. But what they had introduced in this third paper was the idea of enabling characteristics, which are consequences of aberrant conditions of neoplasia that provide the means by which cancer cells and tumors can adopt these functional traits. What does all that jargon mean? Basically, what they explained was that while the core hallmarks that we talked about, take for example, sustained proliferative signaling. That is a that by itself is a capability which is considered to be transformative in nature, right? That it is going to lead to a cancerous condition. However, an enabling characteristic basically means that that it is providing a way by which cancer cells can adopt that functional trait. So enabling characteristic is helping the cancer cell acquire the core hallmark so that it can become cancerous in nature. So it enables neoplasia. That is basically the definition of the same. So that's what you saw here. These enabling characteristics were two. One was non-mutational epigenetic reprogramming. If you remember, we had talked about epigenetic reprogramming a lot. I think uh, at some point of time we talked about it when we discussed different proteins which can be used for inhibition or which can be used for cancer as cancer drugs, basically, that you disrupt the transcriptional circuitry and you disrupt the epigenetic regulation, and by that you can basically target cancer. And apart from that, another concept that was initially that has been proposed as an enabling characteristic are polymorphic microbiomes. The surrounding microbiota has also been proposed in this regard. And apart from that, these two, which are unlocking phenotypic plasticity and senescence, have been proposed as emerging hallmarks in this particular study. So, yeah, basically, these are very subtle differences. Overall, what remains clear till date is that the hallmark by itself will be a functional trait which will be capable enough of inducing transformation in a normal cell and converting it to a cancerous cell. However, the emerging hallmarks either will have to be, will need uh, more meat before it can actually be considered to be a core hallmark. And an enabling characteristic is also different because it enables the acquisition of the hallmark trait to become cancerous. So, yeah, that is overall the discussion, I think. Yeah, so after this, let me just share it again. After this, we understood the first hallmark, which is sustained proliferative signaling. Under that, we looked at the ability of cancer cells to sustain continuous proliferation. We took the example of EGFR and its mediated signaling and other proliferative signaling and their role in cancer. So we understood how there can be physiological activation or physiological uh, stimuli that aid cell proliferation and cell survival. There can be RTK-driven oncogenic events which drive these processes. By oncogenic events, we meant that there can be mutation in the EGFR protein, as a result of which it becomes insensitive to the binding of EGF towards the cytosolic site, and instead, via some intracellular signaling pathways, this protein becomes activated and that switches on the downstream activation pathway, leading to disease. Then we looked at other proteins, that is via redundant pathway. There can be hyperproliferation. Then we looked at RAS-driven oncogenic pathways, BRAF-driven oncogenic pathways, and JAK-STAT-mediated signaling as well. So oncogenic somatic mutations in these key survival genes, they actually drive the sustained activation of downstream genes. The disruption of negative feedback mechanisms that attenuate proliferative signaling, that is lost. Then excessive proliferative signaling, it triggers cell senescence. Um, Then the second one was about evading growth suppressors. So the first hallmark talked about how cells divide very rapidly, and that is considered to be a key hallmark of cancer overall. But apart from that, the ability to evade a negative checkpost, which is the tumor suppressor within the cell. So the ability to evade this um, not tumor suppressor, growth suppressor for a physiological condition, if you just look at it as a cell, then evading that is a major advantage that is often also acquired by cancer cells. So somatic mutations in these genes, they block the feedback loop, which is necessary for cell division to occur in a very controlled, very regulated manner. And secondly, activation of EMT program to trigger invasion and metastasis is another thing that happens. Activation of autophagy does pretty much becomes an advantage in context of cancer as well in the long run. So these are the different mechanisms we had talked about. The evasion of these tumor suppressor proteins. Its role is executed either at a genetic or an epigenetic level, all of which help in aiding the effect of the mutation within the cells. So yeah, then we talked about EMT, whose discussion was done later on. We'll come to it. Then the third hallmark we talked about was resisting cell death. So, autophagy is a cellular degradation pathway for clearing damaged proteins or superfluous proteins and organelles. The recycling of these constituents also serves as an alternative source of energy during the metabolic stresses to maintain homeostasis and visibility. Autophagy often also allows prolonged survival. In contrast to autophagy, necrosis is an uncontrolled form of cell death that results in the swelling of the cell organelles, plasma membrane, rupture, and eventual cell lysis. So that's what you see, a comparison between a normal cell, an apoptotic cell, a necrotic cell. The key difference being the fact that this apoptosis is occurring in a very controlled, very regulatory manner. So it is more of an implosive reaction versus an explosive reaction, much like necrosis. So yeah, these were all of the different sub-mechanisms which help this process. I will skip over it, but you can read it in your own due time. Then we talked about enabling replicative immortality. So are cancer cells immortal? That was the question asked. So Leonard Hayflick, he discovered almost 65 years ago, he discovered that cultured normal human cells, uh, they have limited capacity to divide. So after which they are considered to become senescent in nature, which is known as Hayflick's limit. The two barriers to proliferation, they are senescence and apoptosis. They have actually been rationalized as crucial anti-cancer defenses as well. So senescence is a stage where a cell is not proliferative, but it remains viable. And the reason why replicative immortality overall can actually be enabled is by overexpression of the telomerase gene, which helps in replenishing the lost DNA in every round of cell division, and instead it helps in aiding replicative immortality. Then we discussed very briefly about angiogenesis. So angiogenesis was the process by which new blood vessels are developing or can develop from existing capillaries, which all together they create a complete, regular, and very mature vascular network. And there's the activation of an angiogenic switch, which occurs during this process of tumor progression. It remains on. It causes normally quiescent vasculature to very randomly form blood vessels, and these are not formed in a regulated, in an organized manner. And instead, what happens is that this sort of abnormal sprouting of blood vessels, like you can see that it has formed in a way that it just supplies blood and um, oxygen via blood basically to the tumor. So it has these uncontrolled, unregulated, unplanned way of growing. And this tumor vasculature, it lacks hierarchy. It is very disordered. It is very leaky in terms of its functionality as a as part of the vasculature of that particular individual. So you can see that the outgrowth of blood vessels around the tumor is occurring in this haphazard fashion. So that's what we see here. The tumor secretes VEGF. VEGF increases the expression or expression of genes necessary for blood vessel formation, and it surrounds the tumor overall. And now the tumor has more and more blood supply, more and more oxygen is released, and the tumor sustains a lot better. And finally, we understood activating invasion and metastasis to be a very key process, which is extremely crucial for the movement of this tumor mass from its primary to the metastatic site. And EMT by itself is considered to be a cellular program where cell and cell matrix interactions undergo changes, undergo remodeling. So as a consequence of that remodeling, these cells, they detach from each other, they detach from the basement membrane, and it promotes or it pushes for the mesenchymal fate instead of the epithelial fate. So EMT gives them more tumor initiating and metastatic potential, and they become more treatment or therapy resistant as well. So you can see this was the EMT progression from the epithelial cells. There is partial change. Eventually, the cells become mesenchymal in nature. However, for MET, the reverse occurs, whereby from the mesenchymal state, the cells are going back to the epithelial state. And you can see there is more and more expression of epithelial proteins as well, like ecadherin, occludins, claudins, integrins, cytokeratins, etc. And yeah, so these are the two sets of proteins which have more role to play. And yeah, pretty much that is all you can see from here. So that is what we had summed up. Um, Cancer as a disease of uncontrolled proliferation by transformed cells subject to evolution by natural selection. Those were the substages we talked about, classification into different categories, and the key hallmarks and their impact upon cancer. So yeah, I will hold off on the questions because I don't have slides on these, like on the seed and soil hypothesis and on the role of autophagy in pro-tumorigenic and anti-tumorigenic fashion. We'll look at that in the next and the final session. This is something we have already discussed. How are they different from the core hallmarks? So any doubts, please, you can unmute at any point of time and let me know. I'm just trying to brush over these topics because this was discussed and there's a lot that has already been spoken about and that has been included as part of the live lectures. But just because we want to sum up the course before going through the individual leftover questions, I think it is a good idea to just, reiterate all of the topics you've already learned about. Okay. Next, the first thing we did was address the difference between leukemia and lymphoma. If you remember, lymphoma was lymphoma was the cancer of lymphatic system that originates in lymph nodes only. Leukemia is cancer of the bone marrow or blood cells in the bone marrow, and that is the umbrella term basically. So leukemia and lymphoma, they are called as liquid cancers, and their common origin is from the WBCs of the immune system. Um, then we understood different key components of the signaling cascades. So first of all, you have a couple of things. What are those? First, you have a receptor at the cell surface. On the receptor, a particular protein, often known as a ligand, it sits on the receptor, and the ligand receptor interaction is then able to drive some intracellular signaling molecules, which activate some target proteins, which activate a couple of cellular responses. These responses include altered metabolism in some way, it affects the cytoskeleton in some way, gene expression, cell division, all of those can also be impacted. So this is the model that operates in context of yeast, and the signaling pathway, the components of the signaling pathway have been shown here for your understanding. We understood different types of cell surface receptors, starting off with the extracellular one, starting off with the transmembrane one. So among the different types of transmembrane receptors, we first learned about ion channel receptors. Usually they're just called as ion channels also in a lot of cases. So you can see that it is obviously, since it is a channel, its folding and its architecture is different from that of other TGM receptors. So you have this population of ions that are able to move past, move through the transmembrane receptor, or you can have the 7-helix membrane receptor or transmembrane receptor also. The example being the the GPCR family of proteins onto which the ligand is binding, or you can have receptor that carries some sort of intrinsic enzymatic activity or enzyme associated receptors like recruited receptors, etc. So these were all some examples. Then we understood oncogenic signaling and its role basically. So a regulatory or a control check that is kept within a cell under physiological condition. Its job is to maintain cellular homeostasis, but we learned that these regulatory networks or these regulatory steps, they include certain feedback loops. Feedback meaning the information that has been obtained as a result of the initial signaling that has occurred. So it takes feedback from that initial signaling, and then accordingly, uh, it regulates whether or not more and more synthesis is necessary. So yeah, so that's what we saw. These three types of loops basically. So you have this feed forward loop where you can find the direct sort of the direct activation of protein Z by protein A, whereas the other chain shows you a stepwise progression of the same. Then a positive feedback loop whereby it shows that more and more synthesis of product Z leads to more and more activation of this particular pathway. So it supports its own increase. Then the negative feedback loop inhibits the same. So these are the control checks that have been kept in place. And within the cell, like you can see that there is extracellular fluid which contains signal molecule, binds to the receptor. Then the signal transduction pathway takes place and it activates some particular gene X. So what goes wrong in cancer? First, there are mutations in it's very commonly seen that there are mutations in the cancer pathway genes, the signaling pathway genes. Secondly, epigenetic changes can occur in the TME or the tumor microenvironment. Third is metabolic reprogramming, and fourth is loss of regulatory control. So all four of these, they go wrong in cancer, and there's a lot more as well, but this is the more generic way of representing it. Then, if you remember, I had shown you this schema to explain to you how much information we already have and how much is, even then, this is not enough. We feel like there's still lack of representation. But despite that, this shows you that all of the information about all of the different types and subtypes of cancer that can that has been put into this graph. So you can see the different propensity of different types of cancers, number of individuals, etc. So this was just for your own understanding, so I'm not spending too much time on it. Um, but yeah, then we first understood the examples of some key pathways in cancer. The first example being the RTK RAS signaling pathway. So under the purview of that, we first understood the basic role of RTK RAS signaling in oncogenesis. So you have your receptor tyrosine kinase or RTK, which is present, and that carries this tyrosine kinase domain on the intracellular side, and the ligand obviously binds on the outside. So after the ligand has dimerized, it is able to bind and it stimulates the kinase activity through this dimerized RTK. Uh, yeah, so basically ligand binds, there is dimerization of the two receptors, and then RTK becomes activated via autophosphorylation. Basically, it transactivates itself, and then upon the phosphorylated tyrosine, you can see a lot of proteins are able to bind, and then that is relayed into the cell interior. So, yeah. Then we looked at more details about this. The first comparison being made for for inactive versus active RTKs. So for active RTKs, you can see different conformations. Okay, I'll keep it to that. But basically, you can see that there are different arrangements within the extracellular domain. But apart from that, what you can also see is that there are several processes which are regulated by these interactions. These include cell survival and metabolism, proliferation, differentiation, cell migration, and cell cycle and control. Now, downstream of this RTK ligand interaction, what happens right after it, you can see that there are different directions within which um, it could progress. So you have your activation of the MAP kinase pathway. You can have the activation of JAK-STAT signaling pathway, or you can have PI3K AKT mediated signaling as well. So these are the different signaling networks, any of which can be um, activated, or combination can also be activated depending on what sort of ligand has initially sat on or has interacted with your protein of interest. So yeah, this was pretty much the same thing, I think, in a little more detail, but you can see all of the different types of ligands. You have death factors, you have survival factors, you have chemokines and other signal transmitters. Then you have growth factors and you have the ECM. So they are all merging in via different pathways, either through the RTK-RAS pathway or G protein or GPCR mediated signaling pathway or PI3K mediated signaling pathway, all of this. So uh, then you have these different ways in which oncogenic signaling is often a consequence of haphazard RTK signaling or RTK-RAS activity. So you can find the result, like this happens as a result of a couple of other things. What does that include? First, there can be overexpression of your gene of interest or of your receptor of interest, which leads to more and more gene amplification, which leads to more and more um, expression, and eventually more and more signaling pathway, active signaling as well. Second, you can have activating mutations in the extracellular domain or the kinase domain, which leads to ligand independence. You can have oncogenic fusions. If you remember, I discussed the example of Tempris to ERK fusion or any gene fusion like BCR-ABL fusion, etc. So that leads to formation of abnormal proteins, half of which will stay localized, the other half can migrate anywhere into the cell. Um, Fourth is ligand dysregulation, either autocrine or paracrine, that results in receptor activation and various cellular responses, including proliferation and aberrant tumor growth. Uh, yeah, this actually showed you pretty much the same thing, I think. Yeah, gain of function mutations, amplification, chromosomal rearrangement, RTK domain duplication, and autocrene paracrene activation between different types of cells. All of these are different ways in which RTK mediated dysregulation can occur. Then we talked about the PI3K AKT signaling pathway. So RTK activation, tyrosine phosphorylation, all of these create. So yeah, let me come here. So you can see that the ligand binds to RTK, leading to its activation. And as a result of that, tyrosine phosphorylation of the cytosolic domain of the scaffolding adapter helps in generating a new binding site for recruitment of PI3 kinase, phosphoinositide 3 kinase to the plasma membrane. That then phosphorylates phosphatidylinositol, basically PIP2 to produce PIP3. Um, yeah, then PIP3 can be dephosphorylated back by P10, a lipid phosphatase, and that basically maintains a regulatory check within the cell. So here you can see that from PIP2, PIP3 can be generated, and within the cell, P10 helps in controlling this regulation, therefore acting as a tumor suppressor, and its mutation is often found to be very commonly reported in many different types of cancers. Yeah. Then BIP3 acts as a second messenger and recruits the serine threonine protein kinase AKT to the plasma membrane, where it gets phosphorylated and activated. And AKT signaling promotes cell survival, cell growth, proliferation, and it is a consequence of several metabolic changes as well. I don't think we went into details about this, but basically, AKT is responsible for certain circuitries or in controlling certain circuitries. Majorly, these are involved in proteasomal degradation and translation as well as cell processing within the cell. So, yeah, I don't think we should go into details about this now, but yeah. I don't think we discussed too much about it anyways. Cancer signaling was becoming a very tough topic to actually cross through, so we kept it limited. So maybe I'll skip that here as well. But yeah, what you see here, it shows you the ways in which AKT is controlling metabolism within the cell, and that involves the role of several transcription factors, starting off with HIF1, MYC, ATF4, SREBP, NRF2, FOXO, etc. However, keep in mind that different types of TFs can have different regulatory actions as well. Like FOXO is responsible for glucose uptake and glycolysis. It is responsible for negatively regulating it, whereas HIF1 is able to actually promote that in more amount. Then you have nucleotide synthesis via MYC, control of nucleotide synthesis via MYC, and ATF4 as well. Then you have SREBP, you have NRF2. All of these are regulating different parts. Yeah, I think that was our discussion till session 2. So maybe I'll just complete discussion till session 3 for today, and then we'll wrap up the class. Any doubts, any queries, something you would want clarified that you had in mind from that point of time? Please let me know.
No.
Okay. Okay. In this class, we majorly looked at like we brushed up the basic topics based on whatever discussions we were having in the live class. We went back a little bit on cell signaling, and instead we took up the key concepts. So I had suggested this textbook to everybody, which was Principles of Biochemistry by Albert Leninger, and I stand by that. Please go through it if you can. First, we had understood the types of like some characteristic features of any signaling pathway. The first of which is, oh, just a second. The first of which is specificity, whereby we meant that there should be a unique ligand and there should be a unique ligand receptor interaction which should be able to drive a particular process. Second, amplification of signals should take place inside the cell, which means that if there is one interaction between one ligand and one receptor, that that information or that stimulus should get amplified many folds so as to ensure that there is like the response to a particular stimulus so that it can catch up with the expected effect. For that, there has to be signal amplification. How is that done? That is done by using enzymes which are able to set up a cascade to do so. So when one enzyme activates another enzyme and so on and so forth, the number of molecules that are affected that keeps on increasing geometrically in as part of the enzyme cascade. So enzyme 1 multiple enzyme 2, enzyme 2 activates several enzyme 3s, and it just keeps on progressing in this direction. Third, we talked about desensitization or adaptation. So the activation of the receptor, it triggers a sort of feedback circuit, like you can see that when there's activation of the receptor, to stop it after some point of time or to desensitize the cell to the presence of this particular signal molecule, what does it do? It shuts off the receptor or it removes the same. For this integration, that means it is sort of a synergy or the summed effect or of two different signals that occurs because of the crosstalk between them as well. Okay, so when two signals have completely opposite effects on a metabolic characteristic, such as the concentration of a second messenger X or the membrane potential, the regulatory outcome results from the integrated input from both receptors. So signal 1 and signal 2, both of them are getting combined, or the effect of those signaling cascades are getting combined to generate the response. Then we understood some different subtypes of these receptors. We looked at gated ion channels. Already I've discussed it. So basically you have a ligand which is able to bind to and then move across that transmembrane ion channel. Then you have receptor enzymes, examples being, of course, RTKs that we discussed a lot in detail. Then I mentioned serpentine receptors. I think we discussed GPCR signaling as well, maybe later on, which was an example of the serpentine receptors or 7-pass transmembrane receptors. Then those with no intrinsic enzymatic activity were also shown. You have adhesion receptors, which are responsible for binding molecules and extracellular matrix, changing conformation, and altering cytoskeletal interactions. Finally, we have steroid receptors, which are nuclear hormones. Basically, they are steroid hormones which are able to move, also function as transcription factors, and they are able to obviously bypass both the cell's outer membrane, the nuclear membrane, and then move into the nucleus. So that is what we saw, that it is regulating the transcription of some genes of interest, and therefore its function is crucial for the cell because it does not depend on any other transcription factors. It is able to do the job by itself. It is also a very potent and very promising drug target. So steroid and other thyroid hormones, retinoids, vitamins, etc., by virtue of their hydrophobicity and small size, they are actually able to sort of diffuse across the plasma membrane of the target cells and then bind to intracellular receptor proteins. So some use cytosolic receptors like cortisol, and some nuclear like thyroid and retinoids. These are all structurally related and they're part of the nuclear receptor superfamily. So that's what you see here. In red, you can see cortisol, which is binding to the intracellular receptor protein. There's a conformation change in the receptor, which is then able to move into the nucleus, sit on the gene of interest, and activate transcription of downstream genes. That is the basic pipeline which is followed. Then we looked at G protein signal transduction pathway. So GPCR signaling pathway, the figure on the left was indicating how all the different types of ligands, you can see so many examples, how all these different types of ligands, they're able to interact with one or many of these four types of G proteins, GS, GI, GQ, and G1213. So stimulatory, inhibitory, and 1213 and GQ was, I think, gustatory in nature. So they are responsible for controlling several different types of pathways altogether, which helped in maintaining organismal homeostasis, embryonic development, gonadal development, or necessary for learning and memory. So, yeah. So they are integral membrane proteins with 7 transmembrane segments. They have binding sites for different types of ligands. More than 907 human GPCRs are found. The Genome Sequencing Project has reported even more, and they are likely to be very good drug targets as well. G proteins are like these are the first components. The receptors themselves are one component. Second component are G proteins, which are trimeric complexes of alpha, beta, gamma subunits. G alpha is a GTPase switch protein. It is attached to the membrane. G alpha is acetylated. G gamma is phenylated. Then we have effector proteins like adenylate cyclase, AC, phospholipase, C, phosphodiesterase, ion channels, etc. And secondary messengers like cyclic AMP, cyclic GMP, DAG, and IP3. Then we had looked at one example. I think, yeah, this was the only example probably we looked at. So here we saw how GPCR signaling exactly works. So you have the transmembrane receptor onto which you can see epinephrine has bound, and E, which is shown here in E. Then this occupied receptor was able to cause the displacement of the GDP, which is bound to GS by GTP, leading to activation. So there's an exchange, GDP-GDP exchange, because it acts as a guanine nucleotide exchange factor or a GEF. Then this activated subunit is able to move and bind to adenylyl cyclase AC, and then activates it. What does that do? That leads to that catalyzes or that triggers the formation of cyclic AMP, which acts as a secondary messenger, which activates protein kinase A, which generates the cellular response to epinephrine. However, a negative check or a regulatory control is put on this particular process by the use of the enzyme known as cyclic nucleotide phosphodiesterase. Phosphodiesterase is cleaving that cyclic AMP molecule and liberizing it to generate 5' AMP. But then that is again available for cyclization by AC to generate more and more secondary messenger when required. Apart from that, this was one example. The other example is of phospholipase C, which cleaves inositol phospholipid into IP3 and DAG, which act as second messengers. So overall, that is what happens. You have an activated beta gamma subunit left behind, whereas the activated alpha subunit, it goes and binds to the activated enzyme, which causes the generation of more and more intracellular messengers, which act within the cell. This was the example, the other example that we talked about. So here you can see cyclic AMP process being shown. You have your signal molecule binding to the receptor, which leads to activation of the receptor, the activated adenylyl cyclase, activated alpha subunit of the G protein, then which converts protein kinase A from inactive to activated state once cyclic AMP is generated. Then protein kinase A is able to move through the nuclear pore. It is able to activate the target gene. By activating the regulatory protein, it is able to activate the target gene, which leads to more and more transcription. Um, this cyclic AMP is present in all eukaryotic and prokaryotic cells. While the normal concentration is 10 to the power minus 7 molar, it can increase up to 2 orders in response to an extracellular signal, which stimulates it. Then on the right, you see an example of the IP3 DAG generation via phospholipase C enzyme. So in that case, after activation of the G protein, instead of adenylyl cyclase, the GTP bound active G protein, it binds to phospholipase C, uh, which generates PIP2. And from PIP2, you get diacyl glycerol. Diacyl glycerol binds to protein kinase C and opens up some calcium channels. On the other hand, yeah, on the other hand, you can see IP3 stimulated release of calcium from the sarcoplasmic reticulum or endoplasmic reticulum. Mostly for muscles, this is what is observed. Therefore, I said sarcoplasmic reticulum. From there, calcium stores, they open up, more and more calcium is released. Calcium binds to calmodulin, and with that kinase, what it does, it triggers the um, function via which proteins undergo phosphorylation, and therefore the entire circuitry becomes complete. So this is another example via which GPCRs most commonly operate. I think we skipped TP53 overall. What I had mentioned was that it is certainly the guardian of the genome. So the TP53 pathway, its levels in intracellularly are very tightly regulated by MDM2, which ubiquitinates P53, which leads to its proteasomal degradation. So cell stresses like DNA damage, hypoxia, oncogene action, oxidative stress, or other stress, in all of those circumstances, P53 is necessary. And under stress conditions, P53 becomes active and it gets stabilized by undergoing certain post-translational modifications. And this stabilized P53, it forms tetramers within the nucleus, binds to target DNA, regulates gene expression, and controls many different biological processes. So that's what you see overall, that this P53 signaling under normal circumstances within the cell is extremely crucial. Therefore, its loss or its mutation or any sort of alteration for that matter is a major event that very often occurs in cancer patients, whereby they are often called as P53 mutant tumors or P53 mutant individuals. So yeah, they showed you how abundant the TP53 mutations actually are. So you can see missense to be present, I think, in most individuals. Yeah. Then frame shift, non-shift, silent splice mutations, etc. come much later. Um, yeah. Then here you can see the abundance of different TP53 mutations in different tissues and organs, so that you can see in comparison between males and females as well. Um, then yeah, here you can see how mutant 53 basically ruins or basically disrupts the functioning of so many different pathways, indicating that how crucial its physiological activity actually is. So you can see that mutant TP53 leads to altered transcriptional activity via unfolded protein generation, loss of wild type P53 function, dominant negative effects. Then yeah, then we see that for mutant TP53, proteasomal activity is not properly going to function. There can be drug resistance, there can be invasion and metastasis. Apart from that, via PPI, you can see that genome instability comes up, metabolic reprogramming occurs, TME is affected, and tumorigenesis is very common. Yeah, so this is where we ended the third class for discussion. So we looked at the different signaling cascades. We understood how negative regulation is necessary for normal, uh, for proliferation and for normal cellular maintenance as well. How dysregulation or malfunctioning or imbalance in these pathways becomes a key oncogenic event, and that they can be explored as potent therapeutic targets for the clinical management of cancer patients. I had asked people to go and check this out on the pro-tumorigenic role of TP53 signaling. I'll give another hint. Read about it in context of liver and read about it in context of kidney, which means that for hepatic and renal cancers, read about it. Then we'll have the discussion. Then the off-target and non-specific side effects of targeting these signaling pathways. If you were to improve the specificity of these drugs, how could you do so? These were some questions. These last two questions are more, they're just thinker questions instead of being any textbook or even research paper based questions. So you can just think about it, and we'll maybe pose it as a open discussion type of a question in the next class. So yeah, with this, I am done with my discussion. Anybody, any queries, any suggestions for the last session? And then we'll figure out what to do for that session, and then we'll end today's session for now.
In the next session, could you please tell us about the role of non-coding RNAs in the cancer therapeutics? As we have discussed,
Uh, okay, we can have a short discussion. I'm not sure if everybody is familiar with non-coding RNA, but okay, I'll give a brief discussion and maybe, yeah, we can look. Okay, are you talking in context of that non-invasive diagnostics, etc., when we had that discussion?
yes, ma'am. When you
Okay,
were
okay.
teaching about the cancer therapy, like the different kinds of therapies, like
Mm-hmm.
there is also no, role of non-coding RNA
Okay, we can look at
therapeutics.
that.
I,
Sure, we can look at that.
yes,
I'll keep it a little brief because microRNAs and
yeah,
LNC-RNAs, etc., they are by itself, it will be a whole new topic to then unpack. So we'll just learn a little bit about it, if that's all right. I'll introduce the concept.
yes.
Okay,
Okay,
sure.
ma'am. Thank you.
Sure. Any other suggestions? Anything you would like to discuss? Anybody else also, if you have anything to suggest, please let me know. Okay, if not, then we'll end the session for today. Thank you so much for joining. We'll hopefully have the session on Monday itself. It might get rescheduled to Wednesday because I'm not sure if there'll be a meeting. If it's not there, then of course we'll have the default class. In case there's any change, I'll let you know over mail. And yeah, that'll be our final class, and we'll discuss Ankit's query, and then we'll wrap up the rest of the session also. So thank you so much for joining. Please join for the last session as well. And if you know any of the other candidates, please let them know as just to, contribute if they can join in, it would be great. So yeah, thank you and have a good night. I'll end the session for now.
Thank you.